I'm Dr. Kat Vlies, and I'm from Central New Mexico Community College. We're going to talk a little bit about the resting membrane potential of cells, and then we're going to apply that information to our muscle cell, more specifically our neuromuscular junction. Remember, a neuromuscular junction is a synapses between a somatic motor neurons bulb or axonal terminals bulb and a motor end plate of a muscle cell, a skeletal muscle cell with its T-tubules. I'm hoping that you recognize the different parts of the diagram that I presented to you in an earlier video. But let's, like I said, talk resting membrane potential, and I'll abbreviate resting membrane potential with RMP. First of all, a potential is defined as a gradient of electrical charges. And remember that electrical charges are associated with ions. So we have a gradient of concentrations when we're talking about our sodium and potassium and calcium ions, but we can also talk about a gradient of positive versus negative charges. And that's the definition of a potential. A potential is a gradient of charges. Okay, that said, if we look at a cell in the body, it doesn't matter which cell. Every cell in our body is characterized, and I'll just draw a circle, or more or less a circle, to represent a cell. And every cell is characterized by a distribution of charges right across the cell membrane. So if we look just right across the cell membrane, we see that the inside of the cell membrane is actually less positive compared to the outside. So when a cell is at rest, any cell in the body, it is not disturbed at all, it is not being stimulated at all, then we see that the inside is less positive compared to the outside. And notice I'm using minus signs to reflect that the inside is less positive compared to the outside. It is okay to say that the inside is more negative compared to the outside, but just a word of caution here, and that is that on the inside we have positive and negative ions. You see these ion concentrations here for sodium, potassium, and calcium. Those apply over here as well. So we have on the inside sodium, potassium, and calcium. We also have negatively charged ions like chloride, like amino acids, like phosphate. Same ones on the outside. So we have both positive and negative ions inside of a cell, intracellularly, as well as extracellularly. It's just that right across that cell membrane, the ions arrange themselves such that we see that the inside is less positive compared to the outside. And we can measure this gradient of electrical charges across the cell membrane. We can measure that potential. And if we look at the resting membrane potential of skeletal muscles, then we see that it tends to be around -90. And the unit that we use is called millivolts. So normally in electricity, the unit used is volts, but we're dealing with very small structures called cells, so we use the unit called millivolts. A millivolt is 1,000th of a volt. Okay? The minus sign is important because that is representing the fact that the inside of our cell membrane is always less positive compared to the outside. Now, does every cell in the body have a resting membrane potential of -90? Not at all. You will see that neurons have more of a resting membrane potential that is around -70. But those resting membrane potentials can really vary. So when I've been saying that in a muscle cell, even in a neuron, the resting membrane potential gets disturbed, that means that this gradient gets messed up. And this can only happen in excitable cells. that includes any muscle cell, skeletal, cardiac, or smooth, any nerve cell. Those are the only two cell types that can become stimulated. Or another way of saying it is excited. Or yet another way, which we will use very often, is to say they become depolarized. And what does that mean, depolarized? Well, when a cell is at rest, like it is now, where we see this gradient in charges, we say that the cell is polarized. The cell is polarized. Now, imagine that, and I'll use a different color here, imagine that right here a protein is sticking out and it functions as a receptor for our acetylcholine, and maybe there's another one here. So these are receptors for acetylcholine. So let's now say that this is a cross-section of our cylindrical-looking muscle cell that's coming straight at you, and here we have acetylcholine binding. When acetylcholine binds, remember that allows for ion channels to open up in these protein receptors. Anytime something binds to a protein, it changes shape. And therefore, we're going to see that sodium ions will follow their concentration gradients and rush into the cell, while potassium, which is higher on the inside of our cell compared to the outside, you can see that potassium is high on the inside of our muscle cell there, low on the outside, and so therefore potassium will flow outward. And what we find is that the gradient for sodium is much steeper than that for potassium. And consequently, we're going to see much more sodium moving in than potassium moving out. If more positive particles move in than particles move out, what happens to our distribution of charges? Well, eventually, what we're going to see happening is that around the areas where there are receptors with acetylcholine binding, we see that a reversal in the polarity starts to occur. And we see that in these areas where acetylcholine is binding, that the inside compared to the outside, we're always comparing both sides. The inside becomes more positive compared to the outside because we're primarily bringing in positive ions. You'll see that some books only talk about sodium moving in, but actually potassium always moves out somewhat as well through this same ion channel. This process where we see the reversal of the polarity we call depolarization. And as I said earlier, it is a synonym for excitation or stimulation, all of those things. And if we now apply this information of depolarization to our neuromuscular junction, we get to the following. Here, where acetylcholine is binding to its receptors, we're going to see that mostly sodium will start rushing in, with some potassium rushing out, but we see that while most of our cell is still at rest here, all the way down the T-tubules for now, and same over here, and same over here. I don't want to waste too much time doing this, but you would have little pluses all the way on the outside of the cell membrane, while on this side, similar principle, outside of the cell membrane would be more positive compared to the inside, etc., etc. Well, where our ions are moving in all these areas where we have receptors, so sodium is moving in, potassium moving out, according to my diagram here, there we're going to see the process of depolarization occurring. So these areas, or the motor end plate, you could argue, is becoming depolarized. And so you need to have little minus signs now towards the outside. Now, this depolarization is going to get stronger and stronger the more often acetylcholine keeps binding. So as long as action potentials keep arriving from the central nervous system, we're going to see these sodium ions moving in, potassium moving ions moving out, and the depolarization builds up stronger and stronger and stronger. And eventually that depolarization will be strong enough to open up very special ion channels. And these are ion channels that only open up when there's a very strong depolarization. And these ion channels we find all along our sarcolemma and especially down into the T-tubules. And I'm using little V's to indicate that these are our voltage-gated ion channels. Or some people call them electrically gated ion channels. My pens really squeak quite loud today, don't they? What this implies is that they need a very strong change in the voltage of our cell that was at rest, but is now being disturbed, strong enough to where these ion channels will open. And the first voltage-gated ion channels that will open will be our sodium ion channels and then our potassium ion channels, which are also voltage gated. And when this process occurs, where we first see the opening of sodium voltage gated ion channels, immediately followed by the opening of potassium voltage gated ion channels, we are essentially seeing our action potential recreated and moving down our T-tubules. And again, this occurs wherever there are T-tubules and sarcolemma. So when our sodium voltage-gated ion channels open first, sodium is going to rush in, continuing the depolarization. But right on its tail, those sodium voltage-gated channels, will close. The potassium voltage-gated channels will open, and we now see that we're going to restore our resting membrane potential. So the opening of these sodium voltage-gated ion channels is going to spread the depolarization to deep within the T-tubules. But right on its tail of depolarization, we're going to see that repolarization will occur. So depolarization occurs with the help of our sodium voltage-gated channels. Repolarization, meaning going back to resting membrane potential, occurs with the help of the opening of potassium voltage-gated channels. And how is it that these potassium voltage-gated ion channels get us back to resting conditions? Well, that is because potassium is higher in concentration on the inside compared to the outside. So when we open channels for potassium, potassium will rush out and take with it all of those positive charges. So this is then where we see our action potential very rapidly spreading down into our T-tubules. So we had an action potential occurring here in the nervous system. Then we saw neurotransmitters being released in response to that. So we have a chemical signal being passed on between these two cells. But it's the binding of that chemical signal to these receptor ion channels that then sets up a new electrical signal. Initially, that electrical signal is not very strong, but then it begins to build up and get stronger and stronger and stronger. And eventually it'll become an action potential because voltage-gated ion channels open. The moment we open the very first voltage-gated sodium ion channel, we have started an action potential. And what is so important about this action potential coming down the T-tubules? Well, it can now trigger the opening of our calcium ion channels in our terminal cisternae, such that all of this calcium stored in our terminal cisternae can leave and go bind to the troponin complexes. So the calcium ion channels here are also triggered by very strong depolarization of the action potential, and those are therefore also voltage-gated channels. Similar principle over here. What triggers the opening of calcium flowing in through these channels is the arrival of the action potential, bringing with it very strong depolarization. We have voltage-gated channels in the membrane of the axonal terminal. We have voltage-gated channels along the sarcolemma down into the T-tubules. So what do we call the ion channels to which acetylcholine binds? Well, those are triggered by the binding of a chemical, our neurotransmitter acetylcholine. And so these ion channels are not voltage-gated. What triggers their opening is the binding of the neurotransmitter. And so we refer to our receptors to which acetylcholine binds as chemically gated channels. And I'm going to abbreviate chemically gated channels. What triggers their opening? The binding of our chemical right here, and our chemical being acetylcholine. What triggers the opening of our voltage-gated ion channels is a strong depolarization, very strong depolarization. All right? When acetylcholine binds, it leads to eventually a strong depolarization. That leads to the opening of the voltage-gated channels, and that leads to our action potential, which in turn leads to the opening of calcium voltage-gated channels in the terminal cisternae, such that calcium can go bind to troponin and make the thin filament slide. We'll stop here because our picture is getting quite crowded. There's a lot more to talk about, and we will do that in the next videos. Thanks for watching. Till next time.